Hello everybody. Previously we discussed double fertilization, in which I explained that two structures are formed in flower after fertilization, that is zygote and primary endosperm nucleus. Today I will discuss what happened to these structures. The changes which occur after fertilization, those are called as post-fertilization changes. So there are four post-fertilization changes which occur in flower. Those we will discuss one by one. First is endosperm formation. Second is development of embryo. Third, seed formation. And fourth, fruit formation. All these changes occur in ovule in the flower. So let's come to first post-fertilization changes, that is endosperm formation. What is endosperm? Endosperm is nutritive tissue which is formed from primary endosperm. Depending upon the consumption of endosperm, seeds are of two types, that are endospermic seeds and non-endospermic seeds. Endospermic seeds are also called albuminous seeds. What are those seeds? In which endosperm persists. It is not consumed by developing embryo. Such seeds are called endospermic and they are specially found in monocots like grasses, maize, wheat, rice. Like popcorn of maize which we eat, that is actually endosperm. Second are non-endospermic seeds. Non-endospermic seeds are those in which endosperm is consumed by developing embryo. So in that case, where food is stored? In those seeds, food is stored in their cotyledons. So those are called non-albuminous or non-endospermic seed. And this is specially feature of dicots like wheat and rice. You have seen that their cotyledons are very much prominent. Embryo develops from single-cell structure called zygote, which was formed during double fertilization. Now, how embryo develops? Zygote undergo mitotic division and form two cells. You can say upper cell is called basal cell or suspensor cell. It is formed towards micropylar end of embryo sac. This is micropylar end. And lower is called terminal or embryo cell, which is formed towards chalazal end. Afterward, these cells start dividing. Basal cell undergo rapid mitotic division. You can see after division, long filamentous structure is formed. This whole structure from basal cell is called suspensor. This suspensor and terminal cell of suspensor is called hostorium. What is the function of suspensor? It pushes the embryo towards endosperm. This is embryo. So that embryo can get nourishment from endosperm. This is suspensor. And what about embryo cell? Embryo cell undergo rapid mitotic division and form two tiers of cell, outer circle and inner tier. In the beginning, embryo shape is globular. This round structure is formed, which is called proembryo globular embryo. Later on, it gets demarcated into different tissues and it develops its part later on and it becomes heart-shaped embryo. In heart-shaped embryo, what are the changes? It develops two cotyledons. This is characteristic feature of dicot embryo. And within the cotyledon is embryonal axis, this. And with time, suspensor get perish and embryo and cotyledon start developing in size. This is heart-shaped embryo. And this is the final structure of dicot embryo, which include two cotyledons, which are food reservoir. And in between the cotyledon is embryo. Embryo will get nourishment from these cotyledons. So this is the final structure of dicot embryo, which will form future plant.